Good morning, students. Today, let me explain you about the reproduction in sponges. These sponges are bisexual animals, or what is called otherwise as monoecious, having both the reproductive organs in the same animal. And though these are bisexual animals or hermaphrodites, they exhibit cross fertilization. You know what is cross fertilization? Cross fertilization is nothing but the process of fusion of gametes produced by two different animals of the same species, that is by male and female of the same species. So in this slide you will be observing some pictures showing the details of reproduction in sponges. Sponges are hermaphrodites producing both eggs and sperms, and sponges reproduce sexually by releasing eggs and sperm into the water from the bigger opening of their body called osculum. And as I said earlier, just few minutes back, they undergo cross fertilization and they produce a free swimming larval stage called diplurella. And in the right hand side of this slide, you will be observing a kind of smoky thing that is coming out of the osculum, which is nothing but the release of eggs and sperm by the sponge, a particular sponge. So coming to the other details of reproduction of sponges, and as I said few minutes back, they reproduce both by asexual and sexual methods. Asexually, they generally reproduce by budding, and here in this picture or the slide you will be observing the buds, the lateral buds or the daughter buds that are being produced by a typical sponge, what is known as cycon. And on the right hand side of this slide you observe that there will be the liberation of egg and sperms by the same sponge, and they are going to enter into the body of the another sponge, where the sperm cell is going to be engulfed by a collar cell, and within the body of that sponge, another sponge, the fertilization takes place, and that fertilization results in the production of a zygote. That zygote develops into an embryo after cleavages, and that cleavage producing embryo, and the embryo undergo some changes which produce a type of larval stage, what is called as amphiblastula larva, which exhibits a kind of planktonic nature. Hence it is described here as planktonic amphiblastula larva. And this larva swims freely for some time and settles and attaches to the bottom of the other surfaces and develops into a new sponge. So the details of reproduction of sponges. So sexually, these sponges are reproducing as well as they reproduce by means of asexual reproduction. And in case of sexual reproduction, most sponges are monoecious, as I said, and both the sexes are occur in the same individual, but there is no self-fertilization because individual sponges produce eggs and sperm at different times. Students, in your earlier classes or in the previous years of your study, you must have heard of a concept what is called as protogyny and protandry, wherein protogyny refers to the process of release of the production of female gametes, and protandry refers to the process of production of male gametes first by a bisexual animal. And as the gametes are produced at different time intervals, self-fertilization is usually prevented, and always cross-fertilization is going to facilitate. And from the evolutionary point of view, this cross-fertilization plays an important role. As you know that, students, cross-fertilization always brings about variations, and these variations are very useful for the process of evolution. And in a sponge body, as you studied in the previous class, the cells called choanocytes lost their collars and flagella and undergo meiosis to form flagellated sperm. And the other choanocytes, like amoeboid cells in sponges, probably undergo meiosis to form eggs. So, both sperms and eggs are being produced by a type of choanocytes. Sperm and eggs are released in sponge through oscula. Fertilization occurs in the oceanic water and planktonic larvae are going to develop. In a few sponges, eggs are retained in the mesophyll of the parent. Sperm cell exit one sponge through the osculum and enter another sponge with incurrent water, that is through canal system. Sperm are trapped by choanocytes and incorporated by the vacuole. In some sponges, early development occurs in mesophyll. Cleavage of a zygote results in the formation of a flagellated larval stage. A larva is an immature stage that undergo a dramatic change in structure before attaining the adult body form. The larva breaks free and water currents carry the larva out of the parent sponge. After no more than 2 days of a free swimming existence, the larva settles to the substrate or the substratum and begins to develop the adult body form. Development of sponge larval stages. Most sponges have a parenchyma larva having a size of about 0.2 millimeter. Flagellated cells cover most of the larva's outer surface. After the larva settles and attaches, the outer cells lose their flagella, move to the interior, and formed choanocytes. Interior cells move to the periphery and form pinacocytes. Some sponges have amphiblastula larva, which is hollow and has half of the larva composed of flagellated cells, and they do have almost the same size as that of the parenchyma larva, that is of about 0.2 millimeter size. And on settling, the flagellated cells invaginate into the interior of the embryo and form the choanocytes. Non-flagellated cells overgrow to choanocytes and form the choanocytes. Now coming to gametes. These gametes are a kind of asexual reproductive bodies having a size of about 0.9 millimeter, and these are resistant capsules containing masses of amoeboid cells. Gametes are released when a parent sponge dies, for example in the winter, and amoeboid cells form a new sponge when favorable conditions resume or return. Coming to asexual reproduction. Asexual reproduction of freshwater and some marine sponges involves the formation of resistant capsule called gametes containing masses of amoeboid cells. When the parent sponge dies in the winter, it releases the gametes which can survive both freezing and drying, and when favorable conditions return in the spring, amoeboid cells stream out of a tiny opening called micropyle and organize into a sponge. As I said earlier, sponges also reproduce asexually by budding, and they also reproduce by fragmentation, that is, pieces break off due to wind action and form a new sponge. So there can also be a mechanical injuries in the sea water or in the fresh water, and due to which they undergo fragmentation and each fragment develops into a new sponge. Sponges can also regenerate or regrow the lost body parts and through mitotic cell division, which is also considered as asexual reproduction, and this property makes spongiculture possible. So asexual reproduction, particularly the process of regeneration, helps the scientists to undertake the experiments, the research experiments, particularly to grow sponges, what is otherwise called spongy culture. The details of asexual reproduction is shown in this slide. First one is fragmentation, and as you see, when individual pieces of sponges that are free floating in the water column come together to form a sponge themselves. And in the middle diagram you see budding. Sponges can grow buds that become detached and form sponges themselves. And on the right hand side of this slide you observe the gamules, and as I mentioned earlier and explained earlier, uh, these gamules are covered by a tough resistant covering and having outer membrane and inner membrane, and between which there will be the impregnation of different varieties of spicules, particularly the monoaxon spicules. And this gamule will have an opening on the anterior end, what is known as micropyle, and inside which there is a cavity which is enclosed by a number of archaeocytes. You know, archaeocytes are the mass of undifferentiated cells which develop into new sponge once these archaeocytes or the amoebocytes are released out of the micropyle, and they develop into a new sponge. So gamules also play an important role in reproduction in sponges, particularly during unfavorable conditions. Sponges have tremendous ability to repair injuries and to restore lost parts, a process called regeneration. So regeneration does not imply entire reorganization of the entire animal, but only of the wounded portion. On the other hand, if a sponge is cut into small fragments, or if the cells of a sponge are entirely dissociated and are allowed to fall into small groups and aggregates, entire new sponge can develop from these fragments or aggregates of cells. This process has been termed as somatic embryogenesis, as the new sponge is going to develop from somatic cells. The process is termed as somatic embryogenesis. So this is the another photograph showing the structure of gamete here, and you see there is an opening called micropyle, and as I said earlier, outer and inner membranes are there, and in the center there will be the presence of undifferentiated mass of archaeocytes. Somatic embryogenesis involves a complete reorganization of the structure and functions of the participating cells or bits of tissues. Isolated from the influence of adjoining cells, they can realize their own potential to change in shape or function as they develop into a new organism. So these are the other pictures, the information related to reproduction in sponges. And as I said earlier, asexual reproduction is through budding, fragmentation, regeneration, and in case of freshwater sponges, it is through gamules formation, budding again, fragmentation, and regeneration. So, in both marine and freshwater sponges, you see there is the same phenomenon of asexual reproduction like budding, fragmentation, and regeneration. However, in freshwater sponges, there is a unique phenomenon what is called as gamule formation. And on the bottom side of this slide, you observe sexual reproduction in sponges shown in picture, and it is the same phenomenon which I explained earlier, that the sperms and ova are released from the body of a sponge through the bigger opening called osculum, and if they unite together they fuse to form zygote, and that zygote develop into an embryo, and that embryo develops into a larva, and that larva swims for a short while and attaches itself into a suitable substratum and develops into a new sponge. So these are the larval stages found in sponges or in poriferans. The two main larval stages, one is amphiblastula larva, another is parenchyma larva, the details of which I already explained you. So in the amphiblastula larva, they have micromere and macromere, just like the blastula of a higher animal, and in the center there will be a cavity what is called as blastocele, and on the periphery of this upper part of this amphiblastula larva you come across with some cilia like or flagella like projections, and it is going to swim for short while and then it settles down and develops into a new sponge. And on the right hand side you observe the parenchyma larva having a number of flagella on the upper surface or the outer surface, and in the center there will be the inner cell mass. So this is all the details of reproduction in sponges, students. So let me stop at this level and continue the details in next class. Thank you.